Hi, everybody. I'm Beth. I'm from the UC Santa Cruz Genomics Institute, and like Mo said, I'm just going to highlight some example use cases where you can do data analysis on the AnVIL right now using a lot of tools and tutorials that we've tried to help compile and set up for researchers. I mainly work with the AnVIL and Biodata Catalyst cloud initiatives to try to bring research use cases to these ecosystems. I work a lot with researchers or different types of groups that want to showcase their science, and I'd also be really excited to work with any of you if you're interested in showcasing what you're trying to do with the rest of the community. So I know yesterday you all got an overview of the AnVIL ecosystem. It can be quite complex, and so I wanted to try to break it down from the end user perspective. Really what it's about is that you're trying to bring data and tools into workspaces where you can conduct analyses and make scientific discoveries. So the data can be hosted in a lot of these different platforms that interoperate with one another. So I know there's a lot of tools that are being thrown out, and there's future tools that are coming as well. And so I wanted to go over that a little bit from this type of visualization, where currently data in the AnVIL ecosystem is hosted by Terra. In the future, it will also be hosted within Gen 3, and this can help make that Gen 3 will index the data and make it easier to search across, and also to generate synthetic cohorts. And so data can be easily handed into Terra workspaces from either of these two platforms. And then once you have your data in your workspace, you want to conduct an analysis. And so the AnVIL and ecosystem is trying to bring in tools that are relevant to the scientific community and make them easily interoperate with the data and workspaces in the cloud. So currently, users can search and hand off tools from DocStore, which you'll learn about a little bit more today, right after I present. Luis is going to go over DocStore. There's also the Broad Methods repository and Galaxy, which will be coming in the future. And so all of these repositories of tools should easily interoperate with these workspaces and with the data in the workspaces to help researchers do scientific analyses. So just some links to help you find these data tools and workspaces. Again, you've seen the AnVIL portal in this the presentations yesterday. You can also bring your own data to the ecosystem to use these tools and workspaces. We're going to actually have a tutorial coming soon to help really break it down, how to bring data into the AnVIL and how to potentially sometimes combine it with other data models that might be available from AnVIL or other resources. Then we have DocStore, which we'll present more on. I'll go into it a little bit today. If you're unfamiliar, it's a registry of Docker-based tools and workflows. So these are helping researchers make more reproducible tools and workflows that easily work in the cloud, including with the AnVIL ecosystem. One of the nice features of DocStore are organizations and collections, and I'm going to kind of use this feature to highlight some of the use cases today. Organizations are a feature that allows something like the Broad Institute to host collections of workflows that they create on the DocStore, so you can vet that these workflows were provided by different platforms that you're familiar with. And then collections are basically a way to organize workflows that interact with one another or are different steps in a pipeline. Also there's a lot of really useful workflows available in the Broad Methods repository. So again, the AnVIL ecosystem is trying to bring as many repositories in as possible so that the user has access to many options. And then finally, for workspaces, Terra has a very nice feature called, or featured workspaces, and these are basically tutorials that include example data within them. And they can cover, in addition to workflows, Jupyter notebooks, which provide interactive analyses where you can get the data ready for input into a workflow, for example, or you've already run workflows and you want to do some downstream interactive analyses. The workspaces are also really nice in that they host the workflows and provide the example parameters that you might set up, and they're set up in a way that interacts with the Jupyter notebooks. So it's a really helpful full picture of how to do analyses in the cloud in the AnVIL. So I'm going to highlight a few use cases that are available for you all today. The first one I want to go over is genome-wide association studies. This is a snapshot of a collection of GWAS workflows in the AnVIL organization page on DocStore. And so in this collection, there's a couple workflows available for GWAS. There's simple file conversions to take your BCF into a GDS format so that you can use it with R packages. And then there's a single workflow for performing single or aggregate variant association tests using Genesis, which is R-based. These workflows were contributed by Jamboree, who works at the TopMed Data Coordinating Center. So this pipeline was set up to work with TopMed data. There's a lot of assumptions you might make based on the type of data that you're using. And I think that this pipeline is very applicable to CCTG data as well, because I know that there's a lot of TopMed and CCTG comparisons. And then the Docker-based WDL was developed and is currently maintained by Tim Majarian. And he's also created some really helpful tutorials to do a GWAS in Terra. And another thing I wanted to point out, you can't see it on here, but a lot of these workflow collections come with a markdown, and they can tell you more about the workflows. And in many cases, they might also link to a tutorial where you can actually use these workflows or learn to use these workflows. So what will happen is in that link, you can see that there's actually a Terra-based tutorial for performing genome-wide association studies. This tutorial uses open access data. It's actually 1,000 genomes data that the genomic data has been downsampled, so it's much smaller and easier to use in a tutorial format. And it's paired with synthetic phenotypes. So these are phenotypes that Tim generated that are quite similar to the type of phenotype data you might receive from the TopMed program. And the really nice thing about this is it walks you all the way through a GWAS. So there's interactive Jupyter notebooks, and these notebooks can help you learn more about the cohort that you are using for your analysis. So you can do phenotypic distributions that you can help learn how to pick out which covariates you might use in your analysis. And it also uses the Hail package, which is a Python library that can use the Spark cluster. The Spark cluster works to help parallelize your analyses in the Jupyter Notebook. And in this way, you can explore genetic structure in your cohort and do things like generate a genetic relatedness matrix or filter your variants appropriately for the analyses that you want to do. And this notebook sets the user up to directly input the files that you create in this notebook into workflows that will actually perform the association tests. And again, these were contributed by Tim Majarian and Elisa Manning at Massachusetts General Hospital and the Broad Institute. And in these tutorials and also on the DocStore webpage, there's oftentimes a lot of there's the contact information for the researchers that are developing these tools that you can reach out to and learn more about what they've created and questions that you might have about how to use it for your own use case. Another use case I wanted to highlight that's quite relevant is workflows that are available for phylogenetic analysis of viral genomes, including COVID-19 genomes. There's a collection from the Broad Institute Viral Genomics group on DocStore, and these workflows provide a lot of different helpful tools, including fetching data from things like the short read archive, or generating an assembly and doing this either de novo or with a reference available, performing alignment on the assembly that you generate, and then building phylogenetic trees, leveraging NextStrain to look at the source of these COVID-19 viral genomes. And when you're done with these analyses, for example, if you're bringing your own data into this analysis, you can actually package your data up for submission to GenBake. So this is the collection available on DocStore, which is also used in Terra's featured workspace for doing phylogenetics of COVID-19. This workspace helps you explore the spread and evolution of the COVID-19 outbreak. There's some figures here to show you again that it's leveraging these workflows that are hosted in DocStore, and you can bring your own private or public data into this analysis. The data that's available in the Terra tutorial includes over 300 high-quality genomes from the Massachusetts General Hospital, in addition to over 3,000 samples that are pulled from GenBake from other sources. And there's just a link here to a press release where you can learn more about this. And again, I wanted to just highlight the researchers that are helping drive this, including Daniel Park, which is part of the viral genomics group at the Broad Institute. Another analysis I thought might be interesting to you all are some workflows for single-cell transcriptomics. Again, this is the AnVIL collection on DocStore, or the Cumulus collection, which is in the AnVIL organization on DocStore. And these Cumulus workflows can help you generate counts matrices, demultiplex hashed nuclei. So in addition to single cell, you can also do single nuclei transcriptomics. And then there are workflows to help actually do different types of analyses. I've linked also to the Cumulus documentation. They have a really nice website. And again, there's tutorials here that you can learn to use these workflows in a Terra tutorial. And these were contributed by Bo Ling and Yiming Yang, which are part of the Cumulus team at the Broad Institute. And they're very excited to work with researchers on using the Cumulus workflows. So feel free to reach out to them. Just a highlight of their Cumulus tutorial on Terra. They reproduce in this tutorial, they're reproducing a figure from their Nature Communications paper. And the steps that you can walk through in this tutorial are going from FASTQ files to a normalized counts matrix, learning to do differential expression analysis as well as clustering analysis, and lots of different ways to visualize your data with different algorithms. So you can find a lot of interesting code here that might help you with your own particular analysis. I also wanted to highlight an NIH Interoperability Example. This is where AnVIL and other NIH initiatives are trying to go in the future. And so I wanted to highlight one that is potentially available to you all today. So NHGRI, its cloud initiative is called the AnVIL, and NHLBI, the National Heart, Lung, and Blood Institute, has a similar initiative called Biodata Catalyst. And they use similar platforms that you're learning about today, and they have the same model of an ecosystem of connected tools and platforms that allow the researcher to do their work in the cloud. So the example I wanted to highlight is, so from NHLBI, the Biodata Catalyst currently hosts TopMed data, and this is hosted on the Gen 3 platform, whereas NHGRI, the AnVIL that you're learning about today, hosts data on Terra and currently has CCDG and CMG data. And then I just also wanted to point out, you could also bring your own data to this type of analysis. And so you can easily hand off data from all of these sources to Terra workspaces. There is one complicated factor in that the Gen 3 hosts this data in a different type of data model than Terra does. So combining these two data models in a Terra workspace is complex but doable. And in the future, we're working to try to make this easier on the end user and allow the end user to make these very large synthetic cohorts that can help enable new scientific discoveries. So Terra is the workspace platform for both AnVIL and Biodata Catalyst. So you can import data from different sources into the same workspace, and you can bring tools easily from DocStore and conduct scientific discoveries. So one example I wanted to bring up that you can do today is, say, if you want to do structural variant calling with both TopMed and CCDG data, you could bring both of these data sets into an AnVIL workspace, for example, if you had permission to use both of those data sets in that way. And what some available workflows that we already have for structural variant calling include the VG tool. So this is the variation graphs toolkit from UC Santa Cruz Genomics Institute. So the variation graphs allow you to leverage long read data with short read sequences. So you can build a new graph with a long read data and then map your short reads to that. The graphs also are quite remarkable in that they can hold diversity from multiple individuals. So your reference is not biased to one individual, but can hold variation in this graph format. And so that can allow for better mapping and new discoveries. A tutorial is coming in Terra soon on how to use the VG tools. And another exciting thing I wanted to point out is that these tools are going to be really useful for the Human Pangenome Project. If you're not familiar, the Human Pangenome Project is trying to expand and diversify the human reference by doing reference level sequencing of 350 individuals that come from very diverse populations across the world. And you can, the AnVIL project is hosting this data, and it is all open access data and will be open from the first time that they deposit data in the cloud. And that is scheduled to happen soon in 2020. So you can have access to this data. And in 2020, this project is also hoping to generate some of its first references, I believe, by sequencing individuals from the 1,000 Genomes project that have already contributed their data. So it's, again, a very exciting tools and data that are coming to the AnVIL ecosystem. And with that, that's all I had to say.